to the week 4 live practice session for the NPTEL course on applied environmental microbiology. So like the previous sessions, this session is also a question answer based session, so I hope we'll be having few more participants joining us soon. So the week 4 course content predominantly deals with microbial ecosystem. uh, yes, so the week 4 course content is about microbial ecosystem, and hence we will be again uh, solving some questions on this topic only. So before starting with the session, if you have any particular doubts from uh, week 1 to 4 course content, or any doubts from the previous sessions, you can post it in the chat box. So in the meantime, I'll also post the first question for today. So the first question is, which statement best describes the ecological evenness? And also, I hope the audio is clear and also the screen is visible. If there is any issue with the screen display or the audio, please let me know. You can just post the option that you think is right in the chat box. So the question is, which statement best describes ecological evenness? answer for this question is uniformity of species abundance. So the reason is that species abundance or the relative abundance, uh, when we say this term, we talk about what are the different species that are present in a particular area and what is the number of each species present. Okay, so in that case, when this is uniform, then it means that we have same number of organisms in each species. So that is what we call it as uniformity of species abundance, and that can be called as ecological evenness. So the distribution or the abundance is even, or each of the species is present in almost the similar or same number. So that is why uniformity of species abundance marks the right answer for this question. Whereas when we talk about the number of species present, it is the account of species richness, and we don't talk about evenness here. And we all know that rate of microbial growth is the speed at which the microorganisms reproduce, and it is irrelevant to ecological evenness. And when we talk about genetic similarity and genetic diversity, we are not uh, again relating it to ecological evenness. So it's the uniformity of species abundance, where there are different species present in almost similar numbers, and therefore we call the system to be ecologically even. Okay, with that I'll pose the next question. You can try answering that. So please try to solve the questions in the chat box so that you also get a practice of solving questions. It's not about being right or wrong. Whatever option you think is the most relevant, you can just post it in the chat box.
வந்து வெஜிடபிள் ரைஸ் உங்க ரைஸ் டிஸ்கிரிப்ஷன் சாப்பிடல.
guesses for this question? Remember what a Shannon index is? So, Shannon index usually accounts for both species richness and species evenness. And here we are telling that the richness is constant. Any guesses for this question? Anyone wants to take a guess? Okay, so the answer for this is the species abundance became more balanced. The reason is that when we talk about the Shannon index, it takes into account both species richness and species evenness. Okay, so species richness is the number of different species that are present in the particular area, and species evenness is how evenly they are distributed. The number of individuals that are present in the species, how uniform they are. So that was the first question that we saw. So that was about species evenness. Again, here Shannon index takes into account both richness and evenness. So in our context, species richness is remaining constant, and we have an increase in Shannon index, which means that there is an increase in species evenness. So there are two things in Shannon index: species richness and species evenness. So when we tell that the richness is constant but still the Shannon index is increasing, then it obviously implies that the species evenness is the one that's going to be increasing. Okay, so what do we call species evenness as? It means that the number of different individuals present in a species are almost same. So in that case, the species abundance is going to become more balanced for us to claim that the species evenness has also increased. So in all the other three cases, the given criteria of species richness remaining constant is not followed, or the Shannon index is going to decrease. So when we talk about rare species disappearing, or dominant species increasing, then the evenness is going to be kind of collapsed, right? So in that case, your Shannon index increasing and the richness remaining constant, both these criteria are not going to be satisfied when the dominant species are either increasing or the rare species are disappearing. And also, total population decrease can have either effects, like we cannot get conclusive results from the statement which tells us that the total population is decreasing. But when we talk about the option C, where species abundance becomes more balanced, we claim that the species evenness is increasing, and henceforth Shannon index is increasing, and there are high chances that the richness remains constant. And that is why option C is the right answer here. I hope the statements are not confusing and the explanation is clear to you. If you still want me to repeat the explanation, you can just post it in the chat box. And as already told, please try solving the questions. I've just brought in questions for you to solve and try. It's not about being right or wrong. So when you don't answer, I'm not able to understand whether you are getting the concepts, or is it uh, understandable, or are you still confused. I'm not able to make any judgment. So please try to answer in the chat box. And also, if you have any doubts, please post it. And if you want me to repeat also, please let me know. So with that, I'll go to the third question. So operational taxonomic units are primarily used in which of the following cases? Any answers for this question? You can just type in the option that you think is right in the chat box. And I guess the screen and the audio is clear as well. So, at least for that, can you please give a confirmation in the chat box so that I know that whoever is present in the meeting is able to follow me? Uh, can I please get a confirmation in the chat box if the audio is clear and there is a screen shared from my end? Anyone who is following up? Okay, okay, thank you. So, yes, you're all. I guess that you're also able to follow the question and the answer. So, yeah, you can take your time, but try to solve the questions. Okay, so I'll just discuss the answer now. So, when we talk about OTUs or operational taxonomic units, so in terms of microbial ecology, what we do is we try to group closely related organisms into operational taxonomic units when we are not able to precisely identify the taxonomy uh, up to the species level. So, what happens is uh, we are not able to find clearly which organism belongs to which species. That level of resolution is not possible with our analysis, and yeah, this kind of variations we generally account um, more often in case of microbial ecology. So, what we do is we try to group them uh, into like similar functionally related groups called operational taxonomic units. Okay, it's kind of a proxy for biological species. And here what we, so when do we arrive at this is we typically do a 16s rRNA analysis, and we do the clustering of DNA sequences. And in this clustering we find that all the genes are about having a 97% similarity. That's kind of a threshold, but beyond that we are not able to find the differences and group them into different species. So at that point what we do is we try to combine the genetically similar organisms into operational taxonomic units. Okay, so based on that, the answer is that we use operational taxonomic unit when the genetic resolution is insufficient for taxonomic naming. So that is the right answer. When the species identification is very precise, then we can again call them as a species itself. No need of finding a proxy term called OTU and all. And similarly, um, only if they are cultured microorganisms, then it's very easy for us to identify the individual individual species, and that's going to be again species level identification. And again, um, OTUs and phylogenetic trees have, I mean, to use the term OTU is nowhere related to phylogenetic trees. It's about we are not able to precisely identify it up to species level identification. Henceforth, we are trying to group similar gene clusters with a threshold of, let's say, commonly 97 percentage into same operational taxonomic units. And since microbial ecology is one sector which deals with such diversity of microorganisms, we often encounter this term OTU in microbial ecology. Okay, so the next question is, which metric directly compares community composition between samples? So yes, perfectly right. Bray-Curtis distance or Bray-Curtis dissimilarity index is the metrics that directly compares community composition between samples. So here in Bray-Curtis dissimilarity index, what we do is we try to quantify the difference in the community composition between two different samples or two different size, and generally the value is between 0 and 1. If it is 0, the communities are like perfectly identical, both in composition as well as in abundance. Whereas when we have a Bray-Curtis index of 1, it means that they have no shared species at all. So we are going to compare a community in a sampling site A and a sampling site B. If both the sites are having the same community composition, then the Bray-Curtis index is going to be 0, and like there is no difference at all, they are exactly the same. But let's say not even one species is matching between the site A and site B, then the Bray-Curtis index is going to be 1. So between 0 to 1, the distance is going to range, and this is the only metrics that is going to compare the community composition between two different samples or two different size. When we talk about Shannon index, species richness and evenness, Yeah, they tell about some differences in microbes or the microbial species, but they're not going to compare the community composition between different sites. That is the major difference. So let's when we talk about Shannon index, uh, as we have seen in many of the previous questions, Shannon index is going to consider both species richness and species evenness. So species richness, as I would like to reiterate, is the total number of different species that are present in a particular community. And evenness, when we talk about evenness, we try to find the relative abundance, which means the number of individuals present in each species, and like how uniform they are. Let's say all species some 80 to 90 individuals are present, then it has a high evenness. So that is what we call as evenness. And in a community, when we talk about some 100 different species are present, that is the species richness. And Shannon index is going to take into account both the richness as well as the evenness. Okay, but all these three metrics are for a sample that's taken from a particular site, whereas the Bray-Curtis distance or the dissimilarity index is going to compare the communities that are present at two different sites, and henceforth the correct answer is Bray-Curtis index for this question. Okay, now a simple question maybe. Evenness, when the evenness is 1, what does it mean? Not more than a couple of minutes is needed to answer this question, I suppose. Suppose this is a relatively easy question compared to the previous questions. If the evenness of a particular sample is 1, then what is the inference? Any answer for this question? What is the inference when you find out that the evenness is equal to 1? Okay, so the answer is all species are equally abundant. So when we talk about species evenness, let's say in a particular site we have some 10 different types of microorganisms. If there are 20 individuals, 20 number of microbes in all the 10 different types of microorganisms, then we call the evenness to be 1. When, let's say, out of the 10, there are 2 microorganisms which are 50 in number, 2 different types of microbes which are 50 in number, and the remaining 3 are, or the remaining are 10 in number. Okay, then the evenness is going to be very very low because there is dominance of two types. Let's say from A to E we have the microbes. A and B are 50 in number, and C, D, and E are 10 in number. Then the evenness is going to be very very low. But all A to E are going to be present in some 10 in number. Then it means that the evenness is very high and equals 1. So the inference that you get when you see the evenness uh, being equal to 1 is that all the species that are present are equally abundant. There is no dominance of one particular species in the site that you have considered for sampling. Okay, so another easy question, I suppose. The Shannon index formula primarily depends on which of the following? I think we have already seen this in a couple of questions that we have discussed before today. And obviously, if even if you have doubts, it should be between two options. You can easily eliminate two options from the given. So try to be more interactive so that I get an idea of how much you are able to understand. And if you're not able to follow any question or answer, please let me know in the chat box so that I can try to explain it clearly. And only that will solve the purpose of this live session. Supposing that many of you are from some kind of a life science background, biology, biotechnology, microbiology, or biochemistry, and so on, all the previous few weeks would have been relatively easier for you. And ecological concepts we don't very commonly study, so if you find it difficult to follow or understand, please let me know. And for now, try answering this question. this question, we obviously know that the temperature and mutation rate are not going to be the answer for this question. And in previous two or three questions, we saw that the Shannon index depends on species richness and species evenness. So, based on this, which could be the most relevant answer? Exactly. So, relative abundance, which we have been talking about on all the questions that you have solved so far in the name of species evenness, that is what Shannon index primarily depends on out of the given options. Okay, now coming about diversity components, which of the following is not a diversity component? Exactly. Functional redundancy is not a diversity component. So, as we know, richness, evenness, and abundance all talk about the different kinds of species of microorganisms present in a sample, whereas functional redundancy is kind of different type of microorganisms performing a similar function. So, redundant in the sense the function is repetitive, and it's kind of a measure of what kind of role a particular microorganism plays in the particular ecosystem, so that you try to quantify using functional redundancy. When there is functional redundancy, it means that different species or different types of microorganisms are trying to perform or performing a similar function. Why? Because they are present in a similar kind of environment, and henceforth a similar kind of metabolism uh, could be expected. So, and it's not a diversity component as such. So the next question is, which factor primarily determines the vertical stratification of microbial gills in lake sediments? So till now we have talked about all the indices and stuff. Now we are further moving on to the classification, like population, community, gills, and all these concepts. So you can take your time to understand vertical stratification and gills, and the question is particularly based on lake sediments. first of all, are you aware what is a microbial guild? If you're not able to follow what a guild is or what is vertical stratification, please let me know. I'll explain that first and give you some time to think about the answer. Okay, so first of all, a guild is a group of microorganisms or species uh, that has to live in a condition where they use these same kind of environmental resources, uh, which means that the way that they are going to use is also be going to be kind of similar. They're going to have a similar kind of metabolism, and but they are not like taxonomically very related. They need not be taxonomically very related. They can be different groups or different families or microorganisms in terms of taxonomy, but all these different organisms are supposed to live in a similar environment with similar resources that are available for survival. Henceforth, they can have similar kind of metabolic pattern. So this is particularly called as a microbial guild. And here we are talking about lake sediments, uh, and I hope that you understand what are lake sediments. So when we talk about vertical stratification, imagine something vertically and there are different sections of it. So that is called vertical stratification, and each section is going to have a similar kind of a microorganism. So the question basically tries to ask, you imagine a lake sediment, and you imagine a split up vertically, and there you have different kind of, let's say, separations. So in this separation, uh, what is the primary factor that determines what kind of microorganisms lives in each of this segment? Any guesses? You can just give a wild guess if you're not able to logically answer. See, let's suppose this is a surface of a lake, and here we have different segments. Like there is lake sediment here, we have water here, let that be, and the sediment here, and the sediment extends up to here. Okay, vertical stratification means that you have apparently different sections here, and each of this section is going to have a microbial guild. Okay, so this is what we call as guild. Each group of microorganisms present in each of this section, you call it as guild. So one. Okay, it's not that they are of the same species or so, uh, but since they are present in the same level, it means that they have similar resources to have to consume. Okay, and henceforth we are calling it as guilds. So in this context, uh, what is the factor that determines this vertical stratification is that it is the electron acceptor availability. Okay, so the reason is, for any metabolism you need uh, electron acceptors, okay, which is the terminal electron acceptors. And as you know, let's say a lake sediment, in the top part you will have oxygen, dissolved oxygen, but when you go deeper, oxygen is not going to be present. So you know about like aerobic zone, anoxic zone, and anaerobic zone, right? So in top layer you will have oxygen. In that case you will have aerobic organisms, so aerobic respiration is highly possible there. But when you go further, there is going to be this depletion of oxygen, and hence there has to be other elements that can be used as terminal electron acceptors. And to understand this concept, you need to go back and refresh about your electron tower. So we, in one of the sessions previously, we talked about electron tower. So whenever oxygen is present there, you get more energy, more ATP, and henceforth aerobic respiration is highly supported. But when there is no oxygen, next the organism can consume nitrate and then sulfate. So it keeps going. So the stratification is predominantly based on electron acceptor availability. Secondarily, carbon concentration and other nutrients can also contribute to the vertical stratification of microbial gills, but the primary factor that determines this is the electron acceptor availability. So that is where you see a shift from aerobic microorganisms till anaerobic microorganisms, and that goes from top to bottom. So this is based on the electron acceptor availability, which is a term which will act as a terminal electron acceptor, uh, facilitating the respiration process. I hope it was clear. If there is a doubt, please ask. If you want me to repeat also, please let me know. Okay, so now we come to the basics. Which best defines a microbial guild? I have already answered this question while explaining the previous one. So you have to compare and give the best answer for this. Which option from your perspective is the best? Uh, and a quick question, is it very difficult to follow today because I don't see um, active participation from your side like the previous sessions? If it is difficult to follow, please let me know. so we'll not spend much time in this question. Like we saw previously, microbes that share functional metabolism best defines a microbial guild because they are going to share similar resources, which means that the way uh, way in which they're going to exploit the resource is also going to be similar. So they're going to share functional metabolism rather than the same habitat. Okay, so the next question is, why can extreme microbial diversity exist within a few millimeters of a soil? I mean, you consider a depth of few millimeters of soil and you sample out at different sites. You can still find extreme microbial diversity. The microbial distribution is not going to be kind of uniform. And what could be the reason why you find this extreme microbial diversity is the question. Would someone like to attempt this question? Take any guesses? Okay, so the answer for this question is that strong environmental gradients at microscale are the reasons why we find extreme microbial diversity even within few millimeters of a soil. So here the term strong environmental gradients at microscale means that We talk about millimeters of soil, and within this millimeter, there can be very high or sharp gradients of oxygen, water, nutrients, and one more factor, which is pH. So all these factors are going to critically affect the kind of microorganisms that's going to be present in that particular um, location. So when we talk about soil, yeah, it is a habitat, but it's not uniform. So inside soil itself, there are different regions with different oxygen, pH, water, and nutrient availability. So the nutrient diffusion is not like uniform. When there is high nutrient diffusion, it should kindly be uniform. So that's not happening, and there is no like such rapid genetic mutation happening. There is genetic mutation, but it's not very rapid, so that you find such diversity within millimeters differences. Right. So the correct answer here is the strong environmental gradient at micro scale, and you call it niche. So within a soil matrix itself, there can be different niche. So each niche or a microhabitat can have different oxygen availability, water availability, nutrient, and pH. Right. So in that case, that is why you will find a high diversity. Okay, so the next is pertaining to some aquatic system, where why do microbes benefit from attaching to surface in flowing water? All these are very logically answerable questions, so you can just try your hands out. this question is that when microbes are going to attach to surfaces in flowing water, that primarily improves their nutrient capture. So, in moving water, the nutrient availability is not also very uniform. Uh, so, there is constant movement of both the microbes as well as the nutrients, right? So, but the microorganism, when it is attached to something, uh, some kind of a surface, then this allows a steady supply of nutrients, because there are high chances of the nutrient also getting attached to the surface, or some other different diffusional and adsorptive advantages are present, which offers improved nutrient capture rather than the organism being like suspended in the water itself. uh, but yeah, there is no increase in motility, or there is no enhanced gene transfer per se. And also, when we talk about osmotic stress, it depends on the strength of the water basically, or the potential of the water, and not some kind of attachment or anything. So the direct benefit is that when the microbe is attached to something, you the microbe is going to be benefited by improved nutrient capture. So the next question is, microbial succession in microenvironment occurs mainly because of. So microenvironment we have already seen when we saw about that soil question. There are microenvironment, microhabitats, or niche, whatever you call it. When you consider a whole habitat, a very small part of it is going to be the microenvironment. And what is the reason of microbial succession in a microenvironment? Anyone would like to take a guess in the chat box? Okay. Exactly. So when we talk about microenvironments, so first of all, what is a microbial succession? So over a period of time, uh, the community structure is going to change. So in a community, there are different types of microbial species that are going to be present, but over a period of time, the composition is not going to stay the same. It's going There is going to be some difference in that composition. So this process we call it as a microbial succession, right? And this succession is going to be driven by resource competition and environmental change. And this is particular in terms of microenvironment. When we talk about microenvironment, it's a very small confined space, and here when there is going to be a lot of competition among different microorganisms for the same resource, then whichever is the strongest is going to survive, and the rest are going to be left behind. So this creates an important pressure on the microbial succession. So whichever survives is going to be present over a period of time, leaving the rest of them away from the race. So this is one of the important uh, factors. And environmental change, as we saw, pH, temperature, all of it plays a very important role. So resource and environmental change together, whichever organisms are able to tackle it, are going to survive and are going to be present even after the succession. Okay, so the next question is, which statement about microbial mats is correct? And to add a little clarification to this question, so previously we have discussed about microbial guilds, right? So what we saw was a guild is a group of microorganisms that are able to utilize the same resources in a same way. Like they are present in the same environment, so they are subjected to the same resources, and they try to metabolize them in the same way, and this constitutes a microbial guild. Whereas a microbial mat is a system where again you find a vertical stratification, and the entire stratification together you call it as a microbial mat. So previously I just represented a lake sediment system with some different layers, and the entire set with all those layers is going to be called as a microbial mat. So it's kind of multi-layered, and each of the layer is going to have some microorganisms present in it. So I hope now you understand what a microbial mat is, and each of the layer we are going to call it as a microbial guild. Okay, so now you can try to answer which statement about the microbial mat is correct. is that they exhibit layered metabolic organization. So, as I told you, imagine different layers present in a particular location, and each location is going to uh, have some resource which is going to be exploited by a group of microorganisms using similar mechanism. That is called a guild. And all these layers together is known as a microbial mat. And as we saw in that previous question, this kind of differentiation is going to be because of uh, difference in the carbon source concentration, nutrient concentration, electron acceptor availability, and all of this. So, because there are going to be different nutrient and electron acceptor availability, obviously there is not going to be uniform species distribution. The species distribution is going to be completely uneven. So that statement is wrong, and obviously uh, the stratification in itself is based on functional differentiation only. What kind of microorganism is able to utilize what kind of a nutrient or carbon source, and the terminal electron acceptor that it uses, right? So there is going to be functional differentiation, and it is, yeah, it predominantly exists in marine environments, but it does not only exist in marine environments. It can, like we saw the lake sediment thing or the soil thing, so it can exist in lakes, rivers, soil, and many other environments. It's not like exclusive term for marine environments. So in that case, microbial mats exhibit layered metabolic organization. Okay, so the next question is, why are biofilm associated microbes more resistant to disinfectants? is that the biofilm associated microbes are more resistant to disinfectants because there is EPS diffusion barrier. So biofilm obviously is going to consist of extra uh, cellular polysaccharides, which are uh, the kind of substances that are secreted extracellularly by the microorganisms. They are kind of a slimy substances, and microbes tend to attach to these and form a closer community. Okay, so a kind of a substrate is there for the microbes to attach. And whenever we are going to add a disinfectant into the system, the disinfectant has to diffuse through this extracellular polysaccharide layer and then reach the microorganism. So obviously there is going to be mass transfer limitation, and that is what you call as a diffusion barrier. And hence, whenever there is EPS, whenever there is a biofilm associated microbes, this EPS is the reason why they are kind of more resistant to disinfectants, because it creates a mass transfer or a diffusional barrier. Okay, so the next some questions would be fill in the blank kind of a question only, so you can just try it out. A group of microbes belonging to a species is called. I'll just give a couple of minutes for all the fill in the blank type of questions. Right, they are called populations. Good. Methanogens dominate strongly in dash anaerobic environment. Strongly, uh, I know that methanogens are going to be present in anaerobic environment, and all of you will be aware of that. Okay. This is kind of a tricky question, but I just wanted to introduce this concept that methanogens are present in anaerobic environments, and those conditions are kind of strongly reducing environments. Like you talk about a concept called oxidation reduction potential. And yeah, there is one more question that's related to it. It will be easy for you to solve it now. So you talk about oxidation reduction potential. So methanogens are present in low ORP conditions, which means they are reducing conditions. So this is based on the electron acceptor availability. So when you talk about this stratification, guild formation and stuff, you have oxygen in the top layer and there are aerobic organisms present there, right? But when you come to the bottom, you find methanogens and you have a highly reducing environment there. So this concept you have to keep in mind. Okay. Very open-ended question. You can just type out whatever you feel is right. For this, microbial succession results from what? Okay, so as we saw in that question, microbial succession predominantly results from competition for resources. Okay, so that were few fill in the blank questions. So we are now moving on to some assertion and reasoning question. So the first question is the assertion says that the oxygenic phototropes, they're going to use oxygen, they're going to use light. So oxygenic phototropes often coexist with aerobic heterotrophs. Again, aerobic in the sense oxygen is going to be used, but this time it is heterotrophs. The reason is phototrophs produce oxygen and organic substrate. You can give whether assertion and reason, both of it are right, either of it is only right, or no, both the statements are wrong. That also you can give, and maybe you can try to tell whether the reason is correctly explaining the assertion or just both of them are true statements. Understand the statements and then answer. they produce oxygen, aerobic heterotrophs can use that, and because they're heterotrophs, they're also going to use the organic matter that are going to be produced. So that is right. Okay, the next question, the microbial assertion is that the microbial gills always consist of genetically identical organisms, and the gill classification depends on metabolic function. We have extensively discussed about this in this session, so I guess you should be able to answer this question. First, try to confirm whether both the statements are true. difference in a soil, which is called a microhabitat or a microenvironment or a niche, right? So the reason statement exactly is the same, and there is environmental heterogeneity uh, that is present at a high scale in the microenvironments, right? So that statement is true. And the previous statement, the assertion states that the microbial diversity is decreasing with decreasing spatial scale. Spatial scale in the sense, spatial scale is nothing but a region in space. Like you consider a soil, you consider a small part of that soil, that space is called the spatial scale. So when you consider some 10 millimeters of soil and then you consider 2 millimeters of soil, it means that there is a decrease in the spatial scale. Even though you move from a difference of counting the difference of communities or microorganisms from a 10 centimeter soil sample to a 2 centimeter soil sample, there is going to be microbial diversity and it's not going to decrease. So that is the whole point. Even though there is very small spatial difference, the microbial diversity is going to be very high. So the reason statement correctly reflects that idea, whereas the assertion does not uh, reveal that idea, and henceforth the assertion is false and the reason is true. Okay, so the next question is that the electron acceptor gradients control the energy yield in the microbial ecosystems, and different electron acceptors yield different ATP per reaction. So I think we have discussed this question in the previous class also. And this again pertains to that electron tower, so you can think based on that. So here both the statements are right. So the assertion is that the electron acceptor gradients control energy yield in microbial ecosystems. Obviously true. So when we talk about the stratification and all, depending on which element is, or I mean which compound is available as the electron acceptor, from oxygen to nitrate to sulfate, manganese, whatnot. So whatever is present as the electron acceptor, based on that only there is going to be difference in the microbial population. And we saw, like, that is what is the whole concept of electron tower. When the electron acceptor is going to be different, the ATP production is going to be different. So when the terminal electron acceptor is oxygen, you get the um, most number of ATPs. So when it is going to decrease, so you compare aerobic and anaerobic respiration. In anaerobic respiration, the ATP yield is very low, right? Because the terminal electron acceptor is something else there. So that is the whole point. Okay, so next we have a set of match the following questions. So yeah, I think we can quickly go through these questions. I'll just wait for you for a couple of minutes to answer. Again, this is the stratification that I was talking about. So first layer is the surface lake, and then following that is the upper sediment, then the mid sediment, then the lower sediment, and the deep sediment. So A to E is from top to bottom, whatever stratification will be present. You just have to match it to um, what kind of microorganisms will be present there. And this completely, if you just open the electron tower, you will be able to answer this question. And by now, you should be clear that A is going to pair with 4 and E is going to pair with 1. So the surface lake is going to have oxygenic photodrops and the deep sediment is going to have methanisms. And in between, you have upper, mid, and lower sediments where you have to match the sulfate nitrogen ion radiases. So based on um, electron potential, you can match this. B, C, and D to 2, 3, and 5, which should come where. Only that will be the point of doubt or point to think here. Right? B3. Okay, now it is between C, D, and 2 and 5. Can you just try out that as well? Okay, so it's A4 as we saw, B3 as you have rightly said. Upper sediment, uh, so after oxygen, it's going to be nitrate, followed by iron, and then sulfate, and then methanogens will come into the picture. So after oxygen, it's nitrate, and then iron and sulfate. Nitrate of iron and sulfate is the order that you have to remember. Okay, next is kind of, you have to match the different terms with what they let me post the answers. Obviously, same species are termed as population, as you have rightly answered some time back. And ecosystem, it consists of both biotic and abiotic factors. And guild, we have extensively seen that a microorganisms, a group of microorganisms that survive in a similar resources using similar functions. So it's going to be functional group. And community um, is going to be multiple population, and meta community is going to be linked community. Only there you might have had some confusions. Okay, so from this question on, I'll just post the answers directly because we have limited time left. So, okay, this is about this microbial movement as we saw in one question. So, preconditioning here is going to be surface modification or the substratum modification, and adhesion, you all know that the cell is going to attach somewhere, so that is adhesion. And communication is based on cell signaling, like you could have heard terms like quorum sensing, quorum quenching and all. So it's basically cell signaling. There are some signaling molecules that are released owing to which there is communication between different microorganisms. And when the structure is going to mature, it means that the structure is complex, like biofilm formation, where you have complex EPS formed and all. And dispersion, in the sense there is going to be cell release. This question, when you even know the meaning of the term, generally you can just match the answers rightly. Okay, and the next question is about different uh, types of communication or that microbes generally have. So here, competition, whenever there is resource limitation, there is going to be competition. Whichever is going to efficiently use the resource is going to survive, and that is competition. Cooperation, in the sense cross feeding. Like when there is cooperation among organisms, they are going to cross feed among themselves. And symbiosis, again, is an interesting term, where they are going to coexist and benefit each other, and that you can find among the plant root microbes. And succession, as we saw, is the community change over time. So, and then protection, we saw that disinfectant are not going to affect microbes in a biofilm because there is going to be this diffusional or mass transfer information. So protection matches with biofilm shielding here. Okay. So the next few questions is going to be about uh, some problem only. So I'll just post the answers directly here, so you can just try it out. I'll upload the video as well as the PPT. So whichever you are able to access, you can access the video in energy production and the amount of biomass produced. So I just wanted to introduce this concept to you. So based on that, aerobic is going to be the highest biomass producing zone, whereas methylogenesis or anaerobic is going to be the lowest, and in between, as told, is going to be nitrate, iron, and sulfur. So if I have given iron, it should have been in between nitrate and sulfate. Okay. So with this, we have come to the end of this session. I hope you were able to follow the session. If you have any doubts, please post in the chat box, and looking forward to see you all in the next session as well. If you are finding any difficulty, please let me know in the chat box now